Module 9, the TCA cycle. Chapter 20 in your book is consistent with this online module. So the TCA cycle, it's located in the mitochondria and is responsible for the majority of the ATP generated by fuel oxidation. It accepts acetyl-CoA generated from glycolysis that we've already seen, as well as acetyl-CoA generated from a multitude of pathways. There are several cofactors required for the TCA cycle to turn. Thiamine pyrophosphate, FAD, and coenzyme, or pantothenic acid, are all required as cofactors for some of the enzymes in the cycle. Like glycolysis, the TCA cycle is regulated at 3 different intervening points. The regulation of the TCA cycle is regulated according to the energy level in the cell. When the energy level is high, the rate of oxidation decreases. And likewise, when energy level is low, the flux through the pathway is increased. In addition to oxidizing acetyl-CoA, the TCA cycle is required to generate substrates for various other pathways. The intermediates of the TCA cycle are used for heme synthesis, gluconeogenesis, and fatty acid synthesis, as well as amino acid synthesis. These are coupled to what's referred to as anaplerotic reactions, and these are reactions that involve the replenishing of the TCA cycle intermediates. So the energy yielded from one turn of the TCA cycle is the generation of 1 molecule of GTP via substrate level phosphorylation, 3 NADH, and 1 FADH2. So we've just looked at the oxidation of glucose to generate pyruvate. And pyruvate can generate acetyl-CoA. Acetyl-CoA can be generated by the metabolism of many different intermediates. Fatty acids are oxidized to acetyl-CoA. Ketone bodies are oxidized to acetyl-CoA. Amino acids, such as alanine, can be converted to pyruvate and then oxidized to acetyl-CoA, and ethanol can also generate acetyl-CoA. And all of this acetyl-CoA is accepted and oxidized in the TCA cycle. What we haven't talked about here is how pyruvate gets to acetyl-CoA. So we'll look at that enzyme next. So if you remember, in aerobic glycolysis, we ended with the generation of 2-pyruvate. And we need to get from pyruvate to acetyl-CoA for the TCA cycle. The enzyme that's responsible for the conversion of pyruvate to acetyl-CoA is pyruvate dehydrogenase complex. This is in contrast again to anaerobic glycolysis, where pyruvate is converted to lactate. So again, let's look at how pyruvate gets to acetyl-CoA. So the oxidation of pyruvate by pyruvate dehydrogenase complex to acetyl-CoA generates one of our first NADH. This is a highly regulated enzyme. It's activated by NAD and calcium and inhibited by NADH and acetyl-CoA. Additionally, there are also subunits that are kinases and phosphatases that respond to the energy levels in the cell as well. Let's look more closely at the pyruvate dehydrogenase complex. Again, this complex is responsible for converting pyruvate, generated from our cytosolic glycolysis, into acetyl-CoA, which will be further oxidized in the TCA cycle. This complex is part of a large family of alpha keto acid dehydrogenases. This family of enzymes includes two other enzymes you should take note of. The other is alpha ketoglutarate dehydrogenase, which we'll see in the TCA cycle, as well as the branched chain amino acid dehydrogenase, which we've already discussed in week 6. If you remember, deficiencies in this enzyme are responsible for the presentation of maple syrup urine disease. Common to all of these enzymes is the presence of these three subunits, E1, E2, and E3. And it's the coordination of reactions between these three subunits within the enzyme which allows for the conversion of pyruvate to acetyl-CoA through the passing of electrons through each of these subunits. First, you have the pyruvate dehydrogenase carboxylase subunit, and this subunit requires TPP or thiamine pyrophosphate. The second subunit, E2, is a transacylase subunit that binds lipoate. And finally, E3 subunit is a dihydrolipoyl dehydrogenase subunit that binds FAD. The overall reaction schema is shown here, where the conversion of pyruvate to acetyl-CoA to generate this high-energy thioester bond is coupled to the overall reduction of NAD to NADH. The next slide will illustrate the reaction scheme of each of these subunits more specifically. This is a general schematic of the alpha keto acid dehydrogenases. In the case of pyruvate, R is a methyl group. In this first step, pyruvate loses carbon dioxide, and the remaining two carbons are covalently bonded to thiamine pyrophosphate. This is done by the enzyme pyruvate dehydrogenase. If you remember from looking at thiamine as a cofactor in earlier modules, it's the thiazolium ring of this molecule that's critical for this reaction. The ring acts as an electron sink to stabilize the carbanion that forms as a result of proton loss at C2 in the molecule. So the second step requires lipoamide, shown here. And at this point, the 2-carbon unit is transferred from the alpha keto acid dehydrogenase to lipoic acid to generate this intermediate. At this point, the acetyl group attaches to the thioester in lipoic acid, and at the same time, thiamine pyrophosphate is regenerated. This enzyme complex is referred to as dihydrolipoyl transacetylase. In step 3, shown here, the acetyl group is transferred to CoA. This is still a high-energy thioester bond, and it uses the same enzyme as in step 2. The following step is required to regenerate lipoic acid. In step 4 here, the dihydroxylipoyl dehydrogenase reoxidizes lipoic acid. This requires coenzyme FAD. In this reaction, FAD is reduced to FADH, and FADH2 is simultaneously oxidized by NAD. NADH is the overall product of this reaction. The regulation of the pyruvate dehydrogenase complex is illustrated here. It can be regulated by both covalent modification through phosphorylation and through allosteric regulation, which is shown down here. So first, let's look at the role of phosphorylation in regulating the pyruvate dehydrogenase complex. Like all enzymes in the fed state, the pyruvate dehydrogenase complex is active when dephosphorylated. The complex itself is phosphorylated and inactivated by a kinase. This kinase is termed the pyruvate dehydrogenase kinase. The kinase itself is active under conditions when acetyl-CoA is high or energy is high in the form of NADH. When these two compounds are high, the kinase is activated, and this will phosphorylate and inactivate the pyruvate dehydrogenase complex. This should make sense. If acetyl-CoA is high or NADH is high, this would suggest that there is plenty of substrate for the TCA cycle. Likewise, if we look down here, we can see that acetyl-CoA in high levels or NADH in high levels will also allosterically inhibit the activity of the pyruvate dehydrogenase complex. This is another example of feedback inhibition where the product, acetyl-CoA, can inhibit the activity of the enzyme. On the reverse side, the substrate of the enzyme, pyruvate, can allosterically activate the enzyme, as well as NAD+. So high levels of pyruvate or NAD+, as produced by cytosolic glycolysis, can stimulate the activity of the enzyme to increase production of acetyl-CoA. Additionally, the complex could become dephosphorylated, such as under conditions when insulin is high. You can have activation of a phosphatase by insulin or by high levels of calcium, as illustrated here. And this will activate the complex, allowing it to convert pyruvate to acetyl-CoA. Let's think about this again. Under conditions when pyruvate is high, this will stimulate the activity of the complex. This makes sense. We have a lot of substrate, and the complex will be active to convert pyruvate to acetyl-CoA. High levels of pyruvate will also inhibit the kinase. This is consistent. By inhibiting the kinase, we're keeping the pyruvate dehydrogenase complex dephosphorylated and active. Alternatively, if acetyl-CoA is high, this will inhibit the activity of the pyruvate dehydrogenase complex. High levels of acetyl-CoA will also activate the pyruvate dehydrogenase kinase, which will phosphorylate and inactivate the complex further. Hopefully this illustrates how the pyruvate dehydrogenase complex, our enzyme that converts pyruvate to acetyl-CoA, is reciprocally regulated from the pyruvate dehydrogenase kinase.
So again, the pyruvate dehydrogenase complex converts pyruvate into acetyl-CoA, and the complex itself sits in the mitochondrial membrane. Inhibitors or deficiencies in the pyruvate dehydrogenase complex can be life-threatening. For example, arsenic poisoning inhibits pyruvate dehydrogenase complex, as well as inhibiting the electron transport chain. And because of those activities, it can be lethal. So now that we've converted pyruvate to acetyl-CoA, let's look at the TCA cycle. Again, the TCA cycle is involved in the oxidation of acetyl-CoA. The cycle, again, is also responsible for providing precursors to many synthetic reactions. The net result of the pathway is shown here, where you have carbon dioxide generated, 3 NADH, an FADH2 molecule, and GTP through substrate level phosphorylation. Again, this pathway is in the mitochondria. So in the first reaction, we have acetyl-CoA being converted to citrate. The enzyme here is citrate synthase. The synthase catalyzes a condensation reaction in which a nucleoside triphosphate is not used for energy. In this case, we're going from a C2 molecule to a C6 molecule by the addition of oxaloacetate. The delta G for this reaction is highly negative. So this reaction moves in the forward direction quite readily. The energy for this reaction comes from the cleavage of this thioester bond, releasing CoA. The second step of the reaction is controlled by a condatase, and this is an isomerization reaction where citrate is converted to isocitrate. This reaction occurs through dehydration followed by rehydration, resulting in bond rearrangement. The enzyme aconitase requires iron that is complex from the active site for the enzyme. From there, citrate is converted to alpha-ketoglutarate, and this is our first decarboxylation step in the TCA cycle, where carbon dioxide is released. The enzyme here is isocitrate dehydrogenase. Isocitrate dehydrogenase requires magnesium as a cofactor, and this is also our first regulated step within the TCA cycle. We'll look at that in a few slides. Alpha-ketoglutarate, generated via isocitrate dehydrogenase, then undergoes an oxidative decarboxylation by the enzyme alpha-ketoglutarate dehydrogenase. The conversion of alpha-ketoglutarate to succinyl-CoA also includes a decarboxylation, as well as the generation of a high-energy thioester in the succinyl-CoA compound. The fifth step in the reaction involves substrate-level phosphorylation, where we generate our GTP. The energy to generate this GTP comes from cleavage of our thioester bond and release of CoA. The enzyme responsible for this reaction is succinate thiokinase, and again it converts succinyl-CoA to succinate. Succinate is converted to fumarate via succinate dehydrogenase. And if you remember, succinate dehydrogenase is complex 2 of the electron transport chain. So this FAD is fed directly to the electron transport chain. Fumarate is converted to malate in the next step of the reaction via fumarase. In this reaction, you have hydration across this double bond in fumarate to generate malate. Malate is then converted to oxaloacetate by malate dehydrogenase. This step of the reaction also generates an NADH. This reaction from malate to oxaloacetate requires a lot of energy. It has a delta G of 29.7. It's able to move in the forward direction because oxaloacetate is kept at a reduced level within the mitochondria. Oxaloacetate is an intermediate for other pathways, and the low levels of oxaloacetate facilitate this reaction driving towards equilibrium. So those are 8 steps of the TCA cycle. Through those 8 steps, isocitrate dehydrogenase has generated an NADH for us. Alpha-ketoglutarate dehydrogenase has generated an NADH. Succinate thiokinase has
generated GTP through substrate level phosphorylation. We have FAD generated via succinate dehydrogenase, which again is also part of the electron transport chain, and malate dehydrogenase generating an NADH. All of these are oxidized on the electron transport chain to generate ATP. In addition to regulation of the pyruvate dehydrogenase complex that converts pyruvate to acetyl-CoA, there are 3 steps in the TCA cycle that are also regulated: isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, and malate dehydrogenase. So let's stop and ask this question. The oxidation reactions that convert pyruvate to carbon dioxide are specifically catalyzed by enzymes. Which type of enzymes are they? Dehydrogenases. D. This is the correct answer. So now let's look at the regulation of the TCA cycle. Citrate synthase is inhibited through feedback inhibition. So elevated levels of citrate will inhibit citrate synthase. Isocitrate dehydrogenase is inhibited by NADH. High levels of NADH will also inhibit alpha-ketoglutarate dehydrogenase as well as malate dehydrogenase. Isocitrate dehydrogenase can also be activated by ADP and calcium, and alpha-ketoglutarate can also be activated by calcium. ADP is a signal of low energy within the cell, so this would make sense that high levels of ADP would increase the rate of the TCA cycle, such that we could generate more NADH to be oxidized to generate ATP. Likewise, calcium is also a potent regulator, as this is usually released from the sarcoplasmic reticulum during exercise, and we've seen that in the case of muscle glycogen regulation. So anytime there's high levels of muscle activity, calcium can also activate these enzymes. In addition to the TCA cycle providing energy in the form of NADH, FAD, to be oxidized to generate ATP, the TCA cycle plays an important role in the generation of intermediates for other pathways. Citrate, for example, is shuttled out of the mitochondria via the citrate shuttle and is used as a substrate for fatty acid synthesis. Alpha-ketoglutarate is used as a substrate for amino acid synthesis, as well as to generate neurotransmitter. We've seen this in Module 6, where the transamination of alpha-ketoglutarate can generate glutamate, which is essential for neurotransmitting as well as for generating amino acids. Succinyl-CoA is an important intermediate for heme synthesis, and heme is required for hemoglobin to maintain its function. So this is a critical pathway here. Malate can be shuttled out and used as a substrate for gluconeogenesis. Gluconeogenesis is another pathway by which the body maintains blood glucose levels under fasted conditions. We've already talked about glycogenolysis, and we'll touch on gluconeogenesis in the next unit. Oxaloacetate can also be leached out of the TCA cycle and used for amino acid synthesis. So as these intermediates are being pulled out of the TCA cycle for alternative processes, there are something referred to as anaplerotic reactions that refill these intermediates. These reactions are shown here. This first reaction is the primary and probably most important anaplerotic reaction that we'll see. This is the reaction of pyruvate carboxylase converting pyruvate back to oxaloacetate. This enzyme, pyruvate carboxylase, requires biotin as a cofactor. Biotin will bind carbon dioxide as an intermediate, and the activated carbon dioxide is transferred to pyruvate to form the carboxyl group of oxaloacetate. The conversion of pyruvate to oxaloacetate by pyruvate carboxylase is shown here. Again, this enzyme requires biotin. The enzyme activity is stimulated by acetyl-CoA, such that as flux through the TCA cycle increases, the replenishing of those intermediates is accompanied by that cycle. The next reaction shown here is glutamate, which can be transaminated to alpha-ketoglutarate. This is a readily reversible reaction converted by glutamate dehydrogenase, and the direction of this reaction is dependent on cellular needs. Valine and isoleucine metabolism via the branched chain ketoacid dehydrogenase will eventually generate propionyl-CoA that can be converted into succinyl-CoA. Other amino acids can be converted to fumarate and enter the TCA cycle here. And similarly, aspartate can be deaminated and reconstitute oxaloacetate. The two major anaplerotic reactions are that generated by pyruvate decarboxylase and the regeneration of succinyl-CoA through metabolism of branched chain amino acids. This has taken us through the full oxidation of glucose through glycolysis, the conversion of pyruvate via pyruvate dehydrogenase to acetyl-CoA, and acetyl-CoA oxidation in the TCA cycle. We've also seen how NADH and FAD can be used to generate ATP. So we have now seen all aspects of these metabolic pathways. This slide highlights deficiencies that can occur along the way and how that may impact metabolism. So decreased oxidation of NADH or FADH in the electron transport chain can result in the conversion of pyruvate to lactate. Let's think about that. So high levels of NADH regulate the TCA cycle, remember. NADH can inhibit both isocitrate dehydrogenase as well as alpha-ketoglutarate and malate dehydrogenase, such that high levels of NADH shut down the TCA cycle and pyruvate is converted to lactate. In the case of ischemia, or an inhibitor added to the electron transport chain, if you disrupt the electron transport chain, and again, if you inhibit the electron transport, NADH becomes elevated, you again inhibit the TCA cycle at those key regulatory enzymes, and an individual can present with lactic acidosis as a result of elevated lactate. Deficiencies of enzymes in the TCA cycle can also contribute to lactate formation. We'll look at a deficiency of the pyruvate dehydrogenase complex in the next slide. So essentially, it's required that these processes work in unison, where glucose can be oxidized to pyruvate pyruvate to acetyl-CoA, acetyl-CoA through the TCA cycle, and the reducing equivalents to ATP. This is our homeostatic pathway within the body. So just to reinforce again, which is an inhibitor of citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate? Again, this is NADH. This is an important regulator of the TCA cycle. So let's look at an example of pyruvate dehydrogenase deficiency. This is an X-linked trait. A female infant was born healthy with no evidence of physical or laboratory abnormalities. In the following 3 months, she was brought into the hospital several times. It was noted that the infant fed poorly and had general failure to thrive. Venous lactate levels were evaluated after a fast and after a liquid meal containing 40% carbohydrates. Values for lactate at these time points were 1.3 millimoles per liter and 5.9 millimoles per liter respectively, and this is our normal range. So let's see if we can rationalize this data based on a pyruvate dehydrogenase deficiency. So in this infant, lactate levels were greatly increased after a meal containing carbohydrates. So immediately after eating, the carbohydrates are digested to free glucose. And glucose, again, would be normally metabolized to pyruvate, and pyruvate would be converted to acetyl-CoA by the pyruvate dehydrogenase complex. In the case of a deficiency in the pyruvate dehydrogenase complex, pyruvate remains elevated. In this case, pyruvate will be metabolized by lactate dehydrogenase and will generate lactate. This is the mechanism by which a deficiency of pyruvate dehydrogenase complex can contribute to lactic acidosis. So this summarizes the TCA cycle and the oxidation of acetyl-CoA. In the next unit, we'll look at the generation of acetyl-CoA from fatty acid metabolism as well as ketone metabolism. So in summary, the TCA cycle we've seen is a pathway located in the mitochondria that oxidizes acetyl-CoA to generate fuel. It requires thymine pyrophosphate, FAD, and coenzyme A. Its regulation is at 3 critical points, and energy levels, or the levels of NADH, are crucial to the regulation of those enzymes. In addition to generating energy, the TCA cycle is integral to generating substrates for various other metabolic pathways. In doing so, TCA cycle intermediates also have to be replenished, and they're done so through a series of anaplerotic reactions, the most important being the reaction by pyruvate carboxylase to oxaloacetate. For the next steps, you should review the end of chapter questions, retake the module pre-quiz, and take the module 9 evaluation. From here, you can move on to module 10.